Mike has been doing a lot of impressive research. I first heard of his work in the context of an artificial life conference that took place in Montreal several years ago. And it was like the word on the street that something, astonishing, mind-blowing was happening. And it's rare when you hear scientists get really excited about something. So, and the momentum has been growing since. And it's not just empirical work, although that's also a big part of it. There's also a lot of theoretical doors being opened that have been closed for a long time. So it really feels like an exciting moment where the future is not yet set in stone, and we can all participate in shaping the new sciences of life and mind to come. And it's a science of life and mind that says theory and experiment need to go hand in hand. Theory needs to have traction in experiment, and the experiments can't just be, incremental work. We need to be bold. We need to ask the big questions. Time is limited. We need to make the most of the time and the resources that we have. And so let's just be bold and have fun and see where this journey takes us. And I think there's no one else around at the moment apart from Mike, who really is pushing the frontiers in these regards. So I'm very happy for him to join our conference and tell us about his work. Over to you. Thank you.
Excellent. Thank you so much. And really looking forward to sharing some ideas with you. So what I'm going to do today is talk about some model systems and some frameworks that we've created for thinking about various kinds of novel beings in unconventional embodiments. And specifically, I'm going to talk about some things we've learned about how to relate to unconventional beings. And if you want to look up the primary papers, the data sets, the software, everything is available here. And this is my personal blog about what I think some of these things mean. So, we as humans, and this is the sort of standard adult modern human that features prominently in philosophy of mind literature going back a long ways, stands at the center of several continuous series. We, first of all, are all both on a developmental and evolutionary scale of ultimately we come from cells and subcellular components, but also now because of biological and technological progress, we are also in the middle of this continuum where it's going to be increasingly unclear what a human actually is. And I'm not just talking about language models or things like this. We're first going to encounter beings like this. So some percentage human, some percentage cyborg. This is already happening. We need to start developing better frameworks for understanding what it is like to relate to these novel beings. And I think that we as humans, because of our own evolutionary history, have a lot of mind blindness. We find it very difficult to detect and understand unconventional beings, a little bit like what happened with the electromagnetic spectrum. Prior to a good theory of electromagnetism, we had static electricity and lightning and magnets and visible light and so on. And we thought those were all different things. And so the way that people now talk about machines and organisms and so on, like we thought these were sharp categories. But with a good theory, we now understand that all of them are actually different kinds of manifestations of the same underlying phenomenon. And by the way, that while natively we're only able to recognize a very small portion of that spectrum, with appropriate theory and appropriate technology, we can operate along pieces of this continuum that we were completely blind to before. And I think this is how we are with the spectrum of minds, basically. So in my group, we bounce back and forth between biological and technological media, trying to understand what both of them are telling us about the nature of intelligence. And what we try to do is span the spectrum from deep philosophical ideas to eventually runnable computational models, and then basic science done at the bench, and then hopefully eventually biomedical applications. And a number of the things we're doing are moving to the clinic in areas of regenerative medicine and so on. And that is one way that we test our philosophical ideas. We see if they will push forward actually very practical things that everyone can benefit from. And so our goal is to build tools to enable the detection, communication, and ethical interactions with very diverse intelligences. And our favorite model system is a kind of semi-alien one. We don't really have proper aliens, but it's close enough. You know, the way that cell collectives will live in anatomical space, which I'll talk about, is good because on the one hand, they are related to us closely. On the other hand, they are sufficiently different as cognitive beings that it's very hard for people to think about them. So I think they make a good stepping stone towards truly unconventional minds. And you can see how in this paper how these things are now being extended to regenerative medicine and things like that. So the goal of my framework is to be able to think about all these different kinds of beings, you know, familiar creatures and primates and birds and maybe a whale or an octopus, but also some very weird creatures such as colonial organisms and synthetic new biological life forms. I'm going to introduce you to two of them today. AI, of course, whether software or robotic, and then possibly someday aliens, and also some very strange things that I really won't have time to talk to you about today. And of course, we're not the first person to try for something like this. So here's Rosenbluth, Wiener, and Bigelow in 1943, trying to understand how you get from passive matter to a high-level metacognition and so on. And so my take on all of this is here, at least the 1.0 version of it is in this paper. But the key is that we have to move experimental work forward. In other words, it's not good enough that we just have philosophical ideas. We have to ask what practical implications for research and for the real world do these things have? So these are the criteria for what we're trying to do. So the first thing I want to do in the next few minutes is to show you some features of living material with which you may not be familiar. And in particular, to look at how the material of life navigates some very unconventional spaces. So the first thing to notice is that when all of us took this journey from being a single cell, so an unfertilized oocyte eventually becomes something like this, or even something like this, that's going to, like René Descartes here is going to say that he's not a machine and things like that. But notice that this is a slow and gradual process. It's a continuous process. There is no magic lightning bolt that says, you know, you were just physics and chemistry before, but now you have a real mind. And actually, even that's not the end of the story. As I'll show you, there's a couple of other things that can happen. And so what's happening here is that we are traversing a spectrum of persuadability. In other words, the spectrum of techniques that one might use to relate to us. So from the kinds of simple physical systems that can only be rewired at the hardware level, so physical modifications, to things that are cybernetic and maybe amenable to those kinds of techniques of resetting set points and control theory and things like that, ultimately to systems that are amenable to learning and training, and eventually to psychoanalysis and love and friendship and those kinds of things, right? So this is the kind of spectrum that we are traversing. Notice also that the other thing we traverse is a whole bunch of different disciplines. So we start off somewhere in chemistry and physics, eventually systems physiology, then behavioral science, then psychology, maybe a detour into oncology and bioengineering. But while these disciplines are treated as distinct, so we have separate departments, we have journals, we have funding agencies that treat them as distinct, the substrate, I would argue, is quite continuous, that these are all labels that we put on specific areas, and they have specific tools associated with them. But I think treating these things as distinct natural kinds is very limiting for research. It prevents us from discovering symmetries, deep symmetries across this continuum. And so I'm going to show you some of that today. So this is the sort of thing we're made of. Now, this is a free living unicellular organism known as a lacrimaria, but you get the idea. It's a single cell. It has no brain. It has no nervous system, but it has very high competency in its tiny little agendas. It has a tiny little cognitive light cone. It only cares about the things in a very small region of space-time around it, and it is managing those states, but with great competency. But one of the remarkable things about this, of course, it's full of chemical networks. And I think many of us would agree that a creature like this can be rewarded and punished. It has some sort of valence states. It cares what happens next. You know, if you watch a video of a unicellular die, it's very sad, and we can all sort of see that there is a being that comes and goes into existence. But what about the chemical network that makes it up? Do you think you could reward or punish a molecular pathway? Here, most people would usually say you can't. And so now we have to ask the question, what happened from the time that you had a molecular pathway to the time that you had that creature? But I can tell you that if we actually ask the question experimentally, so we don't treat this as a philosophy or linguistics problem, we simply do the experiment, what we find out is that even molecular networks, okay, not cells, not neurons, just molecular networks alone, in fact, even small ones that have 4 or 5 nodes, they can offer at least 6 different kinds of learning. We can find habituation, sensitization, associative conditioning. They can count to small numbers. Just the chemical network itself is capable of doing this, and they have very interesting causal emergence properties. As these networks learn, their integrated emergence actually goes up. It's a remarkable thing. And so even the material of which single cells are made itself already has proto-cognitive competencies. And in our lab, we're using that we're building devices to train molecular networks, to give them specific memories, to actually wipe other memories, because you can now do drug conditioning and things like this in medical context. So, so you see, we are made of cells. Cells are made of these molecular pathways, which themselves can learn and can scale their causal emergence. And that gives the biological material some really interesting properties at the higher level. So I want to show you some examples of that. First of all, this is a tadpole of the frog Xenopus laevis. Here's the mouth, nostrils, brain, tail. And what you'll notice is that we've prevented the primary eyes from forming, but we've put an eye on the tail. Okay, so here's this eye. If you track the optic nerve, you see that it comes out here. It lands sometimes on the spinal cord, sometimes in the gut, but it never goes to the brain. And yet, these animals can see. And the reason we know they can see is because we've built a device that automates their training and testing in visual assays, so visual cues, so they can learn to behave appropriately in visual contexts. And so this is kind of amazing. You might wonder, why is it that an animal with a radically different sensory motor architecture doesn't need new rounds of mutation, selection, adaptation? You don't need any of that. Out of the box, you make this new creature with an eye on its tail. It doesn't connect to the brain, and yet they have functional vision, and they can learn visual cues. So that's kind of remarkable. This on the fly, sort of zero shot learning adaptation. Here's another interesting example of memory moving through a tissue medium. So these are planaria. These are flatworms. You can cut them into pieces. They regenerate. And what we've done is use that same automated device to replicate McConnell's original data from the 1960s, showing that if you train planaria, and we used a place conditioning task for these rough little bubbles here, you train them, then you cut off their head, which contains a true centralized brain. So you cut off the head. The tail sits there not having any behaviors for about 8 days. By then they grow a new brain. And then the behavior comes back, and you see that they actually remember what they had learned. So not only is the information stored somewhere outside the brain, but also it is imprinted onto the new brain so that the behavior can return when the brain directs the motion. So the information is moving in and out of various tissues, being imprinted onto the new brain, right? And this is something that we're very interested in, how information moves through a collective intelligence, because we are all collective intelligences. We are all made of parts, right? Everything is made of some sort of components. And how does information, global information about the needs of the organism or any kind of agent, move through its medium? But there's more to it than just holding on to the memory and bringing it back when you need it. And that is seen in this amazing example of caterpillars turning into butterflies. Now, it turns out that if you train a caterpillar to a new task during this metamorphosis process, they largely dissolve their brain. Most of the neurons are killed off. Most of the connections are broken. A brand new brain is built for a new life of a different kind of organism in a different environment. But if you train the caterpillar, then the moth or the butterfly remembers the original task. And what's amazing here is not just that memories survive this radical refactoring of the memory medium, but it actually gets remapped onto a new architecture, because if you train the caterpillar to crawl towards a particular color to eat leaves, The butterfly doesn't crawl and it doesn't care about leaves. It wants nectar. And so if that memory is going to survive, it can't survive simply by persisting. It has to be remapped. It has to be generalized and remapped into a new world. It's that memory is now living in a completely different cognitive system. So this is the kind of plasticity and the kind of movement of information that in which we're interested. And so biology lives in this multi-scale competency architecture where every level of organization has its own agendas, its own problem-solving competencies in different spaces and at different scales that allow this incredible plasticity and these amazing feats of maintaining and remapping information and being plastic to novel rearrangements, all the things that our technology doesn't yet have. Okay, so this is one thing that I think is very important about our own embodiment, is that we are collective intelligences, and that all the different scales are also different kinds of cognitive beings with whom we can communicate if we have the appropriate frameworks and technologies. And one of the really important things about it is that by embodiment, as we understand now from this multi-scale architecture, can take place in a lot of different spaces, not just our familiar three-dimensional space with which we tend to be pretty obsessed. We as humans are good at, well, we're not great at it, but we're okay at noticing medium-sized objects doing intelligent things at medium speeds in three-dimensional space. But biology has been solving problems and navigating spaces, doing this kind of sensing, learning, decision-making, and then actuation loop in lots of different spaces, long before we had nerve or muscle. So cells navigate a high-dimensional transcriptional space, they navigate physiological state spaces, and my favorite of all, anatomical morphospace. So this is just the space of all possible geometric configurations of a large-scale body. And our favorite model system is groups of cells, so a collective intelligence formed of groups of cells navigating in a context-sensitive way, anatomical morphospace. I'm going to show you a couple of quick examples. The most obvious thing we can see about navigation towards a specific outcome, meaning that what these cells are not doing is simply mechanically following certain local rules of chemistry, and then emergence and complexity just basically describes the fact that something interesting comes out. That isn't really, that kind of open loop model doesn't fit the data. Instead, animals like this, so this is an axolotl, and these guys are highly regenerative. They regenerate their limbs, their eyes, their jaws, the spinal cords, ovaries, and so on. They show you what goal-directedness in anatomical space looks like. So you can amputate anywhere along the length of this limb. The cells will very quickly spring into action. They can tell that they've been deviated from their correct position in morphospace, and they very rapidly grow, and then they stop. When do they stop? They stop when a correct axolotl limb has been completed. So this is an error minimization scheme. It's a homeostatic process. There is a set point. That's they know when they've reached it. When the error is below some acceptable limit, they stop the movement in anatomical space. So what you need to explain this is a kind of homeostatic model. But it's actually much more interesting than that. It's not simply that cells are able to reduce error towards some particular goal state, and we'll talk in a minute about how they remember what the goal state is, but there's more to it than that. Imagine this interesting experiment. This tail gets grafted to the middle of the flank, and over the coming weeks, what happens is that it actually transforms into a limb in place. It remodels into a limb. Now, look at the world from the perspective of these cells up here at the tip of the tail. We are tail tip cells sitting at the end of the tail. There's no damage, there's no injury, locally there's no problem, there's no reason for us to change, and yet we have to turn into fingers. Why? Because much like in cognitive systems and much like in morphogenetic systems, so that's one of the symmetries between them, local order obeys a global plan. None of the individual cells here know what a tail is or what a limb is, but the collective absolutely does. And it can detect the error of having a tail in the wrong location, and the information propagates, the instructions propagate down to the molecular events needed to turn tail tip cells into fingers. So this is exactly what happens to you and I when we wake up in the morning and we have grandiose research goals, social goals, whatever, like these are very abstract things. Ultimately, for you to execute on those goals, they have to make potassium and calcium ions move across your muscle membranes. Your abstract high-level goals ultimately have to make the chemistry dance in your cells so that you can go do whatever your cognitive system is aiming at. So that kind of multi-scale transduction from very abstract high-level goals, such as having limbs in the appropriate places, has to make a functional difference to the molecular biology of the cells here. Okay, so and that's very interesting. So now, in the next few minutes, I want to talk about how we scale. Well, I want to talk about two things. First, how we scale minds. So as we make a collective intelligence from competent subunits, meaning molecular networks, cells, tissues, all of them have various competencies, how we scale that up into a larger scale collective intelligence, and what communication interface could we use to talk to this unusual intelligence? It lives in a weird space that we're not used to imagining, so anatomical morphospace. We don't know how to really visualize, but mathematics can handle it, and that's fine. And so what I want to tell you about is two concepts. One is called the cognitive light cone, and one is the bioelectric interface. And you can see all of this here. This notion that bioelectricity is basically the cognitive glue. Evolution loves bioelectric mechanisms as the cognitive glue that binds cells into higher level decision-making units, just like you and I are more than the sum of our neurons, partly because of the bioelectric networks in our body. So notice that one way to think about selves, different kinds of agents, is as the scale of their biggest goal. So individual cells have tiny little goals, maybe pH, maybe hunger level, something like that. So their homeostatic set points are very small. They enact this homeostatic process, but the set point is tiny. It's a single number, usually in a very small region of space-time. So if you think about the amount of space and time that each goal refers to, individual cells have a very tiny one, but groups of cells come together into a network that has massive goal states. So for example, this kind of thing, again, no individual cell knows what it is, but the collective is storing something like this. It's projecting into a new space. So this is physiological space, this is anatomical space, and the size of the goal, both in space and time, is much larger. And all of that is described here. Our efforts to model how cells come together in electrical networks so that they can store larger set points. Okay, they can remember much bigger, they can remember these incredible grandiose goals that individual cells cannot remember. And you can map out the cognitive light cone in space-time of all kinds of common biologicals, but also perhaps some aliens and some cyborgs and things like that. And just to show you one practical example of how these sort of philosophical ideas become useful, if we understand that individual cells basically join into networks and thus scale up their goal states, scale up the set points that they can represent. So as I call it, their cognitive light cone. Then something that becomes clear is that process should have a failure mode. And that failure mode is called cancer. Basically, when cells disconnect, individual cells disconnect from that electrical network, they then can't remember the goal they were working on. They go back to their unicellular, tiny little goals of mitosis and metabolism and so on. And so that is exactly what we see in cancer cells. And so our model here said that cancer cells aren't more selfish; they just have tinier selves. Okay, the boundary between self and world has shrunk, and the cognitive light cone, instead of being big like this, has become small. Could we reinflate it again? And so we have a whole set of work in the lab where people are, and it's now moving to human patient tissues, where we basically inject oncogenes. They form tumors, but then we don't kill the cells. We don't repair the hardware, meaning the genetic damage. We force the cells into electrical communication with their neighbors, which they lose, by the way. So oncogenic processes, first thing they do is cause the cell to disconnect from the network. If we force connections, even though the oncoprotein is blazingly expressed here in this tadpole, there's no tumor. It's the same animal. There's no tumor. Because if you connect cells to the electrical network, the network will be building the healthy tissues, not doing individual cell behaviors. So here's an example of how a theory of scaling of the cognitive light cone is taking us to specific biomedical applications. So, so what I was telling you is that the actual information that propagates through that network and is computed is electrical, and that's really shouldn't be a surprise, because when we think about brains and this idea, so here's a zebrafish brain in the living state, doing, thinking about whatever zebrafish think about. We have to ask, where did this come from? Okay, evolutionarily, and we know that neurons evolved from pre-neural cells that had all the same machinery. So ion channels, electrical synapses, this stuff is ancient. It goes back to bacterial biofilms, and every cell in your body has this electrical machinery, and in fact, it connects into networks. And so while the networks in your brain spend much of their time thinking about moving you through three-dimensional space, the networks in your body spend most of their time thinking about how to move your and keep your configuration in the right region of anatomical space. And so we've been developing tools. We developed the first tools to read and write the electrical activities of non-neural cells. So this is an early frog embryo, and here the colors are showing us the voltage gradients. You can see all the electrical conversations that the cells are having with each other. And so much like people in the field of neural decoding want to read this and ask, what is this creature thinking about? What are its goals? What are its capabilities? We have been working to decode the bioelectrics of the body, asking, what is it thinking about? I want to show you a couple of interesting things. The first thing is, I want to show you how cells convince each other to do things. So I'm going to play this. I'm going to play this video in a minute. The colors here represent voltage, and these are two frames of this video, before and after. Keep an eye on this cell right here. Okay, so it's blue, so it has its own voltage, minding its own business over here. This guy's going to reach out, bang. That's it. That's all it took. Here's the before. Here's after. That tiny little connection is formed, and that's it. The cell voltage is now just like that of this other mass, and now the cell is going to come back and work with the rest of these cells to build whatever it is that they're building. That kind of thing, being able to communicate to these cells, is important, because we're starting to learn how to do it too. And when we do it, we can inject, for example, ion channel RNA that puts cells in particular bioelectrical states. And the way that that is interpreted by the other cells is as organ identity messages, prompts. So here again is that tadpole side view. This time, here's the gut, here's the eye, here's the brain, the mouth. What we've done is inject into these cells of the gut, we've injected some ion channel RNA that provides a particular voltage pattern. And the voltage pattern says to these cells, build an eye. And that's what they do. We don't micromanage it. We don't talk to the stem cells. We have no idea how to arrange all the different cells in their combination any more than I know how to arrange your synaptic proteins now so that you'll remember what I'm saying. You handle that yourself, okay? I don't have to worry about the biochemistry. And the same thing is true here. Because we're dealing with a very competent material in morphospace, I can give it a very high level message, build an eye, and it will take care of all the molecular details down the line, just as I was showing you a moment ago. Not only that, the material has some awesome competencies. For example, if I only inject a few cells here, right, what they will do is recruit all this other stuff. The blue ones are the ones we injected. They recruit all these other cells to help them build an eye when there's not enough of them. Other collective intelligences do this too. Ants and termites do the same thing. We didn't have to teach them that. We didn't have to force that. The material already does it. So the trick here is to learn to communicate with it, not to micromanage the molecular states, but to send prompts that the material understands and take advantage of it. This is part of recognizing and communicating with a novel being. We have to understand what is the interface, in this case bioelectricity, for communicating with it, and what is the language. What does it understand? What kind of competencies does it have? What kind of prompts can we use to communicate? We've developed some other things, such as being able to rewrite their pattern memories. So again, in this planarian, if you chop it into pieces, you find that every piece remembers how to build a correct worm, one head, one tail. And we asked, how does it know? And it turns out that there's this voltage pattern that we can read out that says one head, one tail. And if we rewrite that voltage pattern to say, no, actually, you should have two heads, then after cutting this animal, they will indeed make two heads. Now, notice that, first of all, what you're seeing here is a representation of the planarian tissue's answer to what does a correct planarian looks like. That is the memory that is used, the set point that is used to guide regeneration. If we say the set point is two-headed, that is what they build. Also notice that this is not a map of this two-headed animal. This is a map of this perfectly normal-looking one-headed creature. That memory is latent. It doesn't do anything until we cut until it's injured, and then the cells consult this pattern and build whatever it says. So this is actually a counterfactual. It's a primitive version of our mental ability to time travel, to remember things that are not true right now, past things or future states that are not true right now. It's a simple version of a counterfactual in morphospace, because this pattern is not true until the animal's been injured, and it is what I will implement in the future if I get injured. Now, I keep calling it a memory because it has that property. If I make two-headed worms, I can then cut them as many times as I want. They will continue to make two-headed animals. Here are the videos of what they look like. Remember, there's no genetic change here. We haven't touched the genome whatsoever. There's nothing wrong with the genetics here. And you can sequence these all day long. You will never find out that they have two heads, because that's not where the information is. It is not in the hardware. It is actually in the remembered experience that these tissues have had by us using ionophores to change the bioelectrical state such that it encodes a two-headed state, and then the information keeps. It keeps, as far as we can tell, forever. Now, the next thing I want to show you is what happens when we go beyond evolutionarily selected forms, and you can take a look at some implications of that for evolution here. In planaria, we can, by disrupting the bioelectrical signaling, we can ask species of planaria to make heads appropriate to other species. So this nice triangular headed guy can make flat heads like a P. felina, round heads like an S. mediterranea, about 100 to 150 million years distance between these and this. It's not just the shape of the head, the shape of the brain and the distribution of stem cells is just like these other creatures. So this hardware is perfectly happy to visit attractors in morphospace. So in anatomical space, there are different attractors corresponding to these other species. We can chase them into these other attractors where they live natively, but we can push them in. But all of these attractors, you can say, were shaped by evolution. In other words, they exist because of a history of selection forces that made sure that these are the attractors that are there, not something else. What happens when we make new creatures that don't have that history of selection? So I want to introduce you to two things. First of all, xenobots. So when we take apical epithelial cells from frog embryos, they don't die, they don't crawl away from each other, they assemble into this little thing. I want to show you what that looks like. Each circle here is a single cell. So this little group of cells, about 20 of them, it looks like a little horse. They don't all look like that, but I think this one is particularly cute. They move around as a collective. They have all these interesting motions. You'll see a calcium flash here when it encounters this larger mass here. So eventually all these cells will get together and they make something like this. These are self-motile. They use cilia to swim through the water. They can go in circles. They can sort of patrol back and forth. They have collective behaviors like this. You can make them into weird shapes, like this floating donut. Here's a xenobot traversing a maze. It moves forward. It's going to take this corner without bumping into the wall. So it takes the corner, and then here it spontaneously turns around and goes back where it came from. They have spontaneous different kinds of behaviors. One of the amazing things they know how to do is called kinematic replication. If you provide them with loose epithelial cells, they will, both individually and collectively, they will push them into little piles. Then they polish the little piles. And because they're dealing with an agential material, not passive matter, but an agential material, these piles themselves become the next generation of xenobots. And guess what they do? They make the next generation and the next generation and so on. So as far as we know, no other creature on Earth uses kinematic self-replication. We've made it impossible for these guys to reproduce in the normal frog-like fashion, but they found a different way to replicate themselves. There's some other curious things about them. When you look at the system, and you might want to ask, what genes do they express relative to what they normally would do in an embryo? Well, we found out hundreds of new genes are upregulated in xenobots. Among them, I'll just show you one example. Among them, a cluster of genes related to hearing. Okay, so perception of sound and mechanical stimuli. Normal embryos don't express these. This is something new that xenobots are expressing, and we saw this and we thought, that's wild. Is it possible that they can respond to sound? So we put speakers under the dish. We provided the acoustic stimulation, and we found out that sure enough, they do in fact react to it. And this is a novel competency. Normal embryos would not be doing this. This is something that only xenobots do. Now, at this point, you might be thinking that, okay, well, maybe this is some very frog specific thing. You know, amphibians are plastic, maybe just a frog specific thing. So I would ask you the question of what would your cells do if I liberated them from the instructive influence of the rest of your body? So I would introduce you to this creature. This is called an anthrobot. It looks like something primitive you might see at the bottom of a pond somewhere, but it is 100% wild type Homo sapiens genome. These are made by taking tracheal epithelial cells from human patients, and through a fairly simple protocol that we have, they self-assemble into this novel little creature that swims around. It doesn't look like any stage of normal human development. It has, these guys have over 9,000 differentially expressed genes compared to what they were doing in your body. So they have a completely different transcriptome. About half the genome is now differently expressed. They have 4 distinct behaviors, and you can draw a little ethogram of transition probabilities, not 12, not 1, 4 specific distinct behaviors. And if you place them on a dish of human neurons that you've damaged by putting a big scratch through them like this, they will form this kind of superbot cluster and start to heal the gap. If you lift it off, this is what you see. They're sitting there within about 4 days. They take the 2 sides of the wound and they heal it. Now, who would have thought that your tracheal cells, which sit there quietly in your airway for decades dealing with mucus and smoke particles and so on, have the ability to form a separate self-motile little creature that has its own transcriptome that's never been seen before, its own set of behaviors, the ability to heal your body, like you can imagine, right? These are personalized. Someday these will be personalized biobots in the body healing you. You won't need immune suppression because they're made of your own cells. There's no weird materials. There's no nanoparticles here. There's no synthetic circuits. We haven't edited the genome. We haven't done anything at the hardware level. These are native plasticities of cells. They're able to do this. So this raises, as we think about these novel beings, right? So just to bring it back to our fundamental goal is to learn to detect and communicate with novel beings. We also have to understand what are going to be the properties of these new beings that have not been here before. You can't say, you know, you can't derive it from selection. You have to ask what are going to be their capabilities. How can we predict them? So, you know, you can say that we know when the computations were done to design a frog, and that was in the millions of years that the frog genome was interacting with the environment, certain selection forces. And so now here's stages of development, and here are some tadpoles. Okay, we know when the computational cost for this was paid. When did we pay the computational cost to design Xenobots? There was never selection to be a good Xenobot. There haven't been any Xenobots. There haven't been any Anthrobots. Where did all those things come from? It's, I think it's not really fruitful to say that, well, at the same time that humans and frogs were selected, well, we also learned to do this, to have these specific properties. I don't think that works. I think we have to do better than that in understanding why those specific transcriptional states, specific physiological states, specific behaviors, where do they come from? Okay, and I think I don't have time to get into all that here, but I'll just tease this that we have a symposium running here, and you can go here and watch a bunch of videos, including my introduction to what I think is going on, where we study what I think of as the latent space of patterns, and these are patterns of gene expression, of physiology, but also of behavior, aka kinds of minds. And I make the argument in that talk that the mind-brain relationship is basically the same as the math-physics relationship. In other words, the world of Platonic facts about the number E and various mathematical objects. It's not just for constraining what happens in particle physics and other physical objects. It also is something that evolution really makes use of and exploits as a kind of free lunch. And some of those are in fact cognitive properties, not just structural properties. So you can see that here. And so the final thing in the last few minutes, I want to show you something that's kind of our most recent work, which is the use of AI for diverse intelligence research. And this is something that spans the spectrum from something that's a tool to something that eventually will be a colleague. And it has to do with engineering in agential materials. One thing that I believe is that there needs to be an impedance match between the tools you use and what you can see. I think that physics mostly sees mechanism because it uses low agency tools, rulers and voltmeters and things like this. If you want to see if you want to see other minds, you have to use agential sensors. You have to use it takes a mind to be able to see other minds, and so so there's this interesting symmetry between the way that agents navigate their world, right? They make predictions, they make observations, then discrepancy drives the change in their self model and the model of the world, and then they make new observations and so on. And I just want to point out that describes equally well the life of an active agent in any kind of environment, but it also describes science. It describes what we do in science. And so here comes the last of the concepts that I want to transmit to you, which is this blurring of the lines between tools, agents, and the task of navigating and understanding our world. So what we wanted to do, and this is a project together with the Bongard Lab at University of Vermont and with Doug Blackiston, who just got his own faculty position at Tufts. He was a staff scientist with me for years. So together, what we've been creating is the first platform for doing closed loop hypothesis and design of biobots. So one way to think about this, it's a system that has an AI component that makes hypotheses about morphogenesis, about what stimuli would have to be given to cells to get them to build specific things. It then carries out those tests by literally stimulating cells and then observing what kind of xenobots resulted. And then it goes back and it revises those hypotheses based on what it saw. So one way to think about this is just as a discovery engine for communication and collaboration with the cellular material. So we started to build this. They have here are the different kinds of stimuli. So they can do it can do chemical stimuli, it can do vibration, it can do bioelectric kinds of things. This is what it actually looks like. So this is not kind of these are not artists' renditions. This is actually something that we've built. And the idea, again, is that the system is going to learn about morphogenesis by doing experiments in a closed loop system. This is what it looks like. It went online about 3 weeks ago, put together finally in our lab. It's starting to do its thing. And what I think this actually is, we call it MomBot because it's a robot for making biobots. We think that what it is an embodied AI. Okay, so it has a software AI system inside, but it also has a body. Now that body does not move around in three-dimensional space. This thing isn't going anywhere in 3D space. But what it does do is it navigates the space of behavior shaping cues that it is giving to the xenobot cells to build and to build new bodies that are used to explore the latent space of possible forms and behaviors. So it's actually navigating morphospace via the xenobots as a front end. They're using the xenobots as a front end interface to study this morphospace. So it has. So here's what the mind looks like. I don't have time to go into details, but it has a system that has multiple concurrent submodules, just like we have. They're all active at the same time, making suggestions for what experiments are going to be done. Some suggestions eventually percolate to the front, and then it actually carries out those experiments. So it has a body, it has a scalable mind, and it has a scalable physical body. This is the various layers you can see. So if you want to understand the kind of cognitive architecture of the system, this is it, because we created it from scratch. It tries to do infotaxis, so it tries to do the most informative experiments it can that will give it the most information about resolving uncertainty. So this is what agents do in their environment. And it even has something else that's important, is that this thing right here, when it's picking up and dropping cells, right? It can actually this tip through electrical sensing can actually feel when it has something and when it doesn't. So it actually has some proprioception. It actually the physical part of its body actually can sense. It has immediate feedback of what it's doing. So okay, so let me wrap all this up. First, just the next steps of what we're doing. So of course we're using it to discover new biobots, forms and behaviors. We're going to be adding some curiosity about finding problems to solve, not just finding solutions that we ask it for, but actually finding problems to solve. And also keep in mind that because this thing gives stimuli to xenobots, the xenobots could in fact modify their own environment by behaving in ways that cause MomBot to do certain things. In other words, instrumental learning, right? So there's this like very complex, potentially very complex loop between the collective intelligence in the living material and in the AI. So I think that, I'm almost done. There are three ways to think about this. First of all, okay, useful synthetic living machines. So environmental cleanup, exploration, in the body repair, like that's pretty obvious. You know, bespoke living machines. More interesting, I think, is the idea that this is actually robot scientists that can help us crack the morphogenetic code to learn what kinds of stimuli allow us to motivate the cells to build things we want to build. So applications in regenerative medicine, birth defects, bioengineering, and so on, towards this notion of an anatomical compiler, being able to create whatever biological form you want by motivating the cells appropriately. But even deeper than that, we think this is really a tool, the first of a tool to communicate with unconventional minds. It really breaks the categories that we've had separating these are tools, these are scientists, this is the space of intelligence, three-dimensional space. These are developmental processes. Where I really think all those categories are doing more harm than good now, and we really have to create tools like this that help us build intuitions and help us understand how to recognize diverse intelligences, how to communicate with beings in other spaces, and how to actually be part of, right? So we, MomBot, and the Xenobots are all together part of a composite new system that has never existed before. And so here are at least two different ways to think about it, and I think they're both right, and there are probably many others. The MomBot is like this middle bow tie node between human scientists and the Xenobots' collective intelligence in morphospace, right? The MomBot is the translator interface between us and the biological. But you could look at it another way and say that actually the MomBot is also an agent using the Xenobot tissue to explore and to live in and to experience the space of possible forms and functions, anatomical morphospace. So there are numerous ways to cut this up between we, the scientists, between the machine that stands in our lab, between the Xenobots and the latent space of possibilities, numerous perspectives of different active embodied agents that are at play here, and this is we really need to begin to get comfortable with that. So the last thing I'll just say is that because of the interoperability of life, this multi-scale problem-solving competency, pretty much, you know, every kind of combination of living material, engineered material, software is going to be a viable agent. It's going to be some kind of a viable embodied mind. It's going to have the benefit of what I think of as patterns ingressing from this latent space. Everything we know about in the biosphere of the Earth is here. It's a tiny little corner of this option space. So cyborgs and hybrids and all kinds of chimeras, we are all going to be living in this world in the decades to come, and we really need to understand how to enter an ethical symbiosis with these beings. Because when Darwin said endless forms most beautiful, he meant this tiny little corner of this option space, but it's a very wide space, and we need to get a lot better at recognizing and learning to communicate with these beings. And so I'll stop here by thanking the postdocs and students who did the work that I showed you, Josh Bongard and his team, especially for that later part. We have lots of amazing collaborators for this work, and have to thank our funders that have supported us through the last 25 years. And three disclosures that I have to do, because these are companies that have spun out of this work that are commercializing some of this stuff. So Fauna Systems, Astonishing Labs, and Softmax. So that's it. I'll stop here, and thank you very much for listening.
Thank you very much. Thank you very much, Mike, for this inspiring lecture. So many topics to think about. The last part is about the projection of the forms into the bioelectric fields. I hope to contribute something on that topic to your symposium. So I'll leave that for another time. But I just one part in the earlier section of your talk about how to convince cancer cells to reintegrate the body. Something that we've discussed before in terms of could something like MDMA, for example, make them more pro-social as well and things like that. But in this case, you're talking specifically about an electric field. And so one thing that caught my attention is that there's a lot of work right now showing the links between cancer and neural cells. So if they recruit neural cells, and then actually somehow that's even enhances their growth. So I wonder if you have any ideas of what's going on there. It seems like what you're saying is that if it joins the electric field of the body, it should stop being cancer, but maybe some of this work could also be pointing in the opposite direction, that if it somehow connects with the neural electric fields, it somehow enhances the how bad the cancer is.
Yeah, so the devil's in the details here. There's a couple of fine points. First of all, neural bioelectricity is quite different from developmental bioelectricity in the sense that it is a rapid spiking. Okay, what I'm talking about are very slowly changing steady state voltage gradients. Neurons also put out tons of neurotransmitters, which in many cases are attractants and things that the migratory cancer cells love. And they also like to crawl down the axons and everything. So there's a number of different things going on here. But specifically joining via electrical synapses, which is I don't think is what's happening when cancer cells crawl in response to neural cues. Joining into large scale networks gets already transformed cells to stop their local cell level amoeba-like goals and to work on morphogenetic processes. And that both there's lots of good work looking at why it is that those individual cells are interested in neural pathways, but when you do connect them to the rest of the cells, they don't do that anymore. And to your first point, right? So really good. So we have all kinds of efforts, and of course the rate limiting step is getting hold of these compounds because many of these are highly regulated. But all kinds of psychedelics and plastogens and things have a huge role to play in softening the priors of cells about what they can and can't do and things like this. I think you're right on the money there.
Okay, thank you for clarifying. So we have time for questions. I see a hand going up here.
Yeah, Mike, this is, of course, very overwhelming empirical evidence, and you're doing excellent naturalized epistemology. Congratulations for that, and a lot of interesting trends. So we have, of course, we have established certain things which are really interesting take-home points for people working in cognitive science. One of them is, of course, that the cognitive mechanisms without brains are possible. And every cell is electrically active, not merely the neurons. And memory is also located, at multiple places and all that. So I see the interesting aspect that the substrate spectrum, I completely accept that, but I have some tension in the sense of, there are architectures, particularly, the structural architectures, which facilitate certain kinds of affordance landscapes, which does seem to indicate that some kind of transition or some kind of a phase space changes seem to be possible. And also the mind blindness that is an important point to talk about, but which seems to be an important condition for the kind of particularity that we have as human beings to analyze things. In that sense, that isn't that actually a competence rather than a blindness, because mind blindness helps us to see specific things. Of course, there are too many points to ponder about, but I'll stop with these two questions.
Yeah. Okay. So first on the issue of continuum versus some kind of phase transitions or something. Look, I'm in principle not against phase transitions. It's fine if you want to make the case for a specific, like here's a parameter, and I no matter how closely I zoom in, you think that at some point there's a jump in capability. I'm not against that in principle. Honestly, though, the examples that I've not seen really good examples of that. what I see when people propose those kinds of things, oftentimes it's because they've chosen a particular scale. And when you zoom in, usually with time, but sometimes also other things, if you zoom in, it actually it looks very continuous. It just looks like a jump when you sort of stand back at human scale. But it's okay if somebody has a good example of that can be defended. That's fine, and that's not incompatible with what I'm saying. Except that one thing that people often do is first of all, they assume that these phase transitions are the null hypothesis, and that if I want to say it's continuous, then that should be argued for. I think it's the other way around. I think evolution and development are telling us that the null hypothesis is continuity, and if you want to make an argument for specific discontinuous phase transitions, you have to make that argument. And the reason that I think that's important is because when people assume that the categories are sharp, okay, and there are, this is what we do in chemistry, and this is what we do in behavior science, and those are different things, and there's a big transition. There's a problem with this, and the problem is that it prevents the porting of tools and hypotheses. So if you believe in these sharp categories by default, meaning without really examining to what extent are they really sharply different, what you will never do is take, for example, the tools of behavioral and cognitive science and apply them to some other domain. Right. And people say this to me all the time. They say, well, it's a category error. Brains do this and that. You can't do this because they have in mind some ancient philosophical category that they're worried about running afoul, and it prevents you from doing new experiments. I treat this all as an empirical task. Take the tools, apply them, see what happens. We can all see it either got you somewhere or got you nowhere, and then we can build a spectrum. So I think it's really important. What the spectrum allows you to do, it allows you to pour tools. And it turns into from a philosophical or linguistic problem, it becomes an empirical problem. And then, sorry, what was that? Just remind me what the second thing was. I forgot what the second question was about.
Just briefly, because we have one more question waiting.
The second question is about the kind of competence that we achieve because of the specificity, mind-blindness, and the light cone.
Yep, got it. Okay, so look, yeah, I agree with you on the one hand that being able to draw, being able to construct categories and to generalize and to make distinctions between things is definitely a competency, right? And it's great that we have it. What I mean by mind blindness is that I think we're prone to ignoring some fundamental symmetries. So for example, it is very difficult for people to like it's all well and good that we can recognize we have some definitions of intelligence and things like that. But as soon as you step away towards a different scale of size or speed or in a different space, suddenly people find it very hard to use those to think about those kinds of things in the same way. And I think that's artificial. I think we have to be able to expand the scale pretty much just like we did for the electromagnetic spectrum. It's not that X-rays and infrared don't have different properties. Of course they do. And as an engineer, you learn to rely on that. But it's still the case that we now understand there's a unification has taken place. We now understand that even though we can't see some of those things, they really are manifestations of the same underlying phenomenon, and that helps us develop technologies to deal with it. And I think that's what's happening here. We're just natively, of course, because every cognitive system has blind spots, and we're just not good natively. That's why we have science and philosophy to help us with that.
Okay, thank you, Mark. Last question.
Can you say a bit more about, I guess, how you understand the role or, yeah, I'm just very ignorant on this particular topic, but is the bioelectricity playing a mediating role? Is it carrying information? What is it about voltage that's interesting to the systems that it's regulating? Just maybe some introductory stuff to that type of thing.
Sure. Okay. The easy thing I can say is that what it does in the body is basically exactly what it does in the brain, which is that the nice thing about voltage is that it is already an example of generalization. So for example, if cells, a group of cells will, first of all, when you respond to voltage, as opposed to responding to specific levels of potassium or sodium or calcium. You have generalized, and there are many different ways to get to the same voltage, and so you've now you're now able to also make bioelectrics makes it very easy to make feedback loops, both positive and negative. It's easy to make memory using bioelectrics. It's easy to scale. So once you start connecting cells, then you start sharing memories. Basically, cells that are connected this way start to they lose their individual identity a little bit because they start to share their memories through the electrical synapses. So it has, and we could go into considerable detail now. We understand quite a bit of this about how the electrical connections are allowing a larger network that can store bigger set points, and that I think is really the key here, where individual cells can represent very tiny little goal states and set points. And we have lots of simulations showing how this happens. The larger electrical network can integrate across both space and time, and it can store larger set points for future behavior. So this is the pattern I am going to make if certain things happen, and they can store that memory. And it's this gigantic thing that individual cells could not keep. So it is a scaling. It is a set of scaling policies. There are others, stress sharing. There are some other things that are not bioelectrics. Bioelectricity is not unique in this, but it is a way to scale goals. And I think scaling goals is basically scaling minds fundamentally. And then it's a whole other thing that we can talk about as far as whether there's a layer above that, which I think there is. But it's a really good set of mechanisms and policies for scaling goal states for goal-directed activities.
Thanks, Mike. I really like the idea that it's a process of generalization at work already, like in this move from biochemistry to bioelectricity. Sounds to me almost the kind of thing that I mean with the absorption operator. It's yeah, I'll think about that a bit more. Great.
Very cool.
So thank you so much for your presentation and for fielding the questions. And
thank you very much.
Thank you.
Thank you very much. I appreciate it.
Thank you. Bye.
Thank you, everyone.